Hello everyone. Today I'm going to demonstrate some new in-progress features for a JBrowse demo involving the multiple sequence alignment viewer and the 3D protein structure viewer. So we're going to look at this online demo, and I'm going to just start by looking up a gene of interest. So we'll navigate to that gene. We'll see the gene structure from the GFF30 file, and I'm going to right click and to start do the launch MSA view. So this gives us a multiple sequence alignment using data from UCSC, which is a multiple alignment of the canonical transcript of this gene against 100 other species. Clicking submit, we now see our multiple sequence alignment with all these different species, and the MSA view plugin allows us to zip around. You can see a lot of data here, but importantly, we can mouse over positions in the MSA view, and you'll see this little yellow bar changing position on the genome. And we can zoom in on the genome all the way down to the base pair level, and we'll see that if I'm highlighting over, say, the letter H here, that's also highlighting the letter H on the second frame of that protein translation, the green H. And that corresponds with the green color of the transcript above it. Again, N, K, H, K, S. So there's good correspondence there. And we can also do the same thing going the other direction, where if I'm highlighting over the L, D, S, K, H, so it has this sort of bidirectional quality. And that's very useful for, connecting your data in the genome space to the protein space and vice versa. And this works for both positive and negative strand genes. And it's been a work in progress doing all those coordinate transforms. But we can also see a similar thing with 3D protein structures. So in the last demo, I was using this data from UCSC. This is data from Biomart that tells us which PDB entry is associated with a particular transcript isoform. So I'll hit submit again. Let's go look at this 3D structure. So very nice. This is, again, still the P10 gene. And we can click and on a particular part of this, and you can see it says we clicked the letter Y of the amino acid single letter code Y, and that is highlighting a Y now on the genome. Again, the blue color is matching up with the reading frame here, so you can do that with any of these letters. We have A highlighting an A, Q highlighting a Q. So that is, again, another really cool way to connect your data in genome coordinate space with, say, important binding regions or chemical properties of the proteins. And as long as I have you here, I'll show you one more kind of cool new development, which is the multiple alignment format viewer in so-called MAF viewer for JBrowse 2. And this is actually a port of a JBrowse 1 plugin that I did recently that lets you look at, again, a big alignment against all these different species, but this is a whole genome alignment instead of just a protein alignment. So you can get a lot of data about the alignment of related species, and this is 30 different mammals, and you can see that right over the proteins, the protein coding regions, there's a high degree of conservation. Then outside of there, it varies much more than that. And you know, you can use this to dig into sort of other cross-reference data sets, like say Cosmic. And one thing I like to do is compress the data visualization down, get more dense, and you can sort of look at any given variant and say, oh, well, you know, there's not any variation across the tree of life there. That could be deleterious to have a mutation there. So this is just some of the recent goings on, and if you have any questions about this, let me know. Thanks for watching.